Hello, good evening, people. Hello, good evening. Hello. Yeah, can you hear me, people? Am I audible? Please send a chat to the main line. Sedum says yes. Okay, nice. So, people, we here to discuss paper 11 today, right? Paper 11. So, can you find paper 11? I have paper 11. I'll send it to the chat now. So, those who don't have the paper 11, what they can do now, neither. So people, we have to discuss these questions. We have to discuss these questions. We'll start with MCQs. We'll start with the first question. Question number 1. People, we have arranged questions from 3 sections. There is enzymes and excuse me 1 minute. The questions come from enzyme lesson. And then plant life cycle. Plant life cycle. Right. So first question regarding enzymes. First question regarding enzymes. Increase the velocity of reactions by reducing the activation energy. Enzymes increase the velocity of reaction by reducing activation energy. Correct, Ned? First is okay, ne? Oh. First is okay. Containing active sites which can combine the activators. Active sites which can combine activators. No, activators usually bind with an allosteric site. Ne? So it's wrong. So it is wrong. And need proteinous cofactors for the catalytic activities. Enzymes need the proteinous cofactors. What do you call proteinous cofactors? Coenzymes, ne? For the catalytic activity, not always, ne? It says sometimes. Most certain, sorry, word certain. Then next one is people alter the nature of the end products. Alter the nature of the end products. No, that's wrong. And then people are being used up. Are being used up during reactions. 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 Wrong, neither. So question number 2. So we have the answer, people. First one is the answer. Question number 2. Which of the following statement is incorrect regarding enzymes? All enzymes are globular proteins. All the most neither wrong. So people, some enzymes can be some enzymes can be RNA molecules. Neither. Some enzymes are RNA molecules. Right? So say taking all enzymes are globular protein is not correct. We can't agree with that. Then second one, some enzymes can alter properties of the end products. No. Presence or absence of enzyme does not alter the nature of the end product. Sir, two mics are working at once. Oh, really? Okay, I know what to do then. Is it okay now? Please send me a message. Is it okay now? Hari there? Yep, ne? Hari, okay. Right, question number 3. Statement 3, always enzyme catalyzed reactions are reversible. Always enzyme catalyzed reactions are reversible. What do you say about that? That is correct, ne? Always enzyme the most of the always the name. Most wrong, ne? Some proteins in the membrane have ability to act as an enzyme. Yeah, that's correct. Membrane proteins can act as enzymes, yes. Enzyme catalyzed reactions can never be altered by external molecules. Enzyme catalyzed reaction can never be altered, no. Activators, inhibitors can alter, neither. So, you know the answer now. Fourth one is the answer. Question number 3. Question number 3. Question 3. Select the correct statement regarding allosteric regulation of enzymes. Regulatory molecules bind to an active site of the enzyme. Regulatory molecule bind to an active site of the enzyme. Regulatory molecule bind to an active site of the enzyme. In cooperativity, it happens. But normally people, they go and bind with the allosteric site. So we'll leave this first one as a question. Binding of regulatory molecules do not affect the shape and the function of the enzyme. Binding of regulatory molecules do not affect the shape and the function of the enzyme. Do not affect the shape and the function of the enzyme. Binding of regulatory molecules do not affect the shape and function of Wrong, neither. They affect neither. Regulatory molecules are always activators. No, inhibitors are there too. Most enzymes regulated by allosteric regulation are made from 2 or more subunits. Correct. ADP function as allosteric activator and produce ATP during anabolism. ADP function as allosteric activator and produce ATP during anabolism. The catabolism, wrong, neither. So we have the answer. I think we can agree with the fourth one. Regulatory molecules bind to an active site of the enzyme, not always. Cooperativity, we can see that happens, otherwise not. Right, so I don't agree with the first one. I'll leave that and I'll go for the best answer. Fourth one, question number 4. Question number 4, correct statement regarding enzymes. Competitive inhibitors to the enzyme are irreversible. Competitive inhibitors to the enzyme are irreversible. Competitive inhibitors to enzymes are irreversible. Competitive inhibitors to the enzyme are irreversible. Competitive inhibitors to the enzyme are irreversible. Competitive inhibitors to the enzyme are irreversible. Neither. Wrong, neither. Competitive inhibitors are reversible, neither. Most irreversible. We'll leave that as a question. When an inhibitor bind to active site, when an inhibitor bind to an active site, it stabilizes the inactive form of enzyme. Inhibitor bind with an active site, it stabilizes the inactive form of the enzyme. Active site neither. Allosteric site neither, am I wrong? ADP function as allosteric inhibitor in some enzymes. Activator neither. Wrong. Activators and inhibitors bind to regulatory sites. Activators and inhibitors bind to regulatory sites via covalent interactions. Activators and inhibitors bind to regulatory sites via covalent interactions and affect enzyme function. Activators and inhibitors bind to regulatory sites via covalent interactions and affect enzyme function. Covalent varadine lamai. We can't take that neither. Then to reverse competitive inhibition, substrate concentration can be increased, correct? So fifth one is okay. Competitive inhibitors of enzymes are irreversible. Nah, neither. So ඒකත් වැරදි නේද? Competitive non-competitive කිව්වේ reversible නේද? Irreversible කියන්න බෑනේ. Wrong නේ. එහෙනම් answer is 5. Answer is 5. Right, fifth question. Which of the following correct regarding allosteric regulation of enzymes? Allosteric regulation of enzymes. These are made up of one or more subunits. One or more නෙවෙයි, two or more වගේ එකක් තිබුණනේ. Wrong. Regulatory molecules affect only the shape. Regulatory molecules affect only the shape of an enzyme. Regulatory molecules affect only the shape of an enzyme. Affect only the shape of an enzyme. Shape, not only shape, the function also. Function also. The activity can be activated by binding one substrate molecule to an enzyme. The activity can be activated by binding one substrate molecule to an enzyme. The activity can be activated by binding one substrate molecule. Okay, again, activity can be activated by binding one substrate molecule to an enzyme. Binding one substrate molecule. Cooperativity ke tanam correct. Regulatory molecules bind to specific regulatory sites of an enzyme is irreversible. Wrong, ne? Intermediate products of metabolism involved in regulation of the production of more end products than required. Intermediate products of metabolism involved in regulation of the production of more end products. Intermediate products kila අපි ගත්තේ නෑ wrong නේද ළමයි. එහෙනම් out of the given answers people, third one is a better answer. Effect of pH on the rate of the reaction of two enzymes A and B is illustrated in the following graph. Effect of pH on the rate of the reaction of two enzymes A and B is illustrated in the following graph. Which of the following statement regarding enzyme A and B is correct? Both enzyme A and B can be functional within a single organ or of an organism. A and B can be functional within the single organ. Now we learn these two. One acts on stomach and the other one is acting on the small intestine. So it's not the same organ. A and B can function within the single organ. Wrong. Enzyme AB could be trypsin and pepsin. AB could be trypsin and pepsin. A, not in order, the A is pepsin. This looks like this order is not correct. A is pepsin. B is trypsin. Respectively, given rate of reaction of enzyme A at pH 1 and pH 3 could be more or less equal to the rate of the reaction of enzyme B at pH 7 to 9. Okay. Optimum pH range of the enzyme A is 0 to 2. Wrong, ne? No, neither. No, we can't agree that. Because this is optimum pH is 2. Optimum pH is 2. Most of the enzymes of man are similar to enzyme B. Similar to enzyme B. Most of the enzymes are having pH range of 6 to 8. So sixth question, we can go with the third one. Ne? Sixth question, we can go with the third. Seventh question, which one of the following statement incorrect regarding factors affecting the rate of enzyme reaction? Factors affecting the rate of enzyme reaction. Factors affecting rate of an enzyme reaction. Enzyme of an organism have almost same optimum temperature. Enzymes of all organisms have almost same optimum temperature. Enzymes of an organism have almost same optimum temperature. Of an organism, correct, neither. Right? Normally, if you take human people, we maintain 37 degrees Celsius body temperature. Ne? So, which of the following statement is incorrect? So, I'll take first one as correct, but I'll come back for that statement once again. Components which are essential for the catalytic activities of certain enzymes are called cofactors. Components which are essential for the catalytic activities of certain enzymes are called cofactors. Essential catalytic activities of certain enzymes are cofactors. Okay, Neil. The rate of the reaction increase until the enzyme molecule will be saturated at particular substrate concentration. Correct, ne? Honey, wrong again, honey. Most of the competitive inhibitors are irreversible. Most of the competitive inhibitors are irreversible inhibitors. Competitive inhibitors are irreversible, wrong, neither. Rate of enzyme reaction increase proportionately with pH and then become constant. Hurry. So we'll take people fourth one as the answer. Fourth is the answer. Which of the correct statement regarding animals of phylum Chordata? Which of the correct statement regarding animals of phylum Chordata? Only animals of class Reptilia possess scales in their body covering. Wrong, neither. Always also, ne? Animals belongs to class Chondrichthyes have cloaca. Chondrichthyes cloaca. Hurry, ne? Animals of the class Amphibia live in freshwater, on land and marine. Animals of class Amphibia live in land, marine, wrong, neither. No marine members. Animals of class Aves, Reptilia and Amphibia possess eggs with shells. Animals of class Aves, Reptilia and Amphibia possess, wrong, neither. Amphibia don't have shelled eggs. Hollow nerve cord is located between the digestive tract and the notochord. Hollow nerve cord is located between the digestive tract and the notochord. No, it's actually present above the notochord. Wrong. So we got the answer. Second is the answer. Question number 5. Which of the question number 9. Question number 9. Which of the following is present only in one class of phylum Chordata? Which of the following present in one class of phylum Chordata? Scales. Scales. Following is present only in one class of phylum Chordata. Scales. One class Gandabani, the amphibian, Aves and Reptilia. Two-chambered heart. Osteichthyes and Chondrichthyes. No. Muscular tail. No. Absence of marine species. Absence of marine species. Oh, correct. Endothermy, no. So we have the fourth one as the answer. Which of the following are common to reptiles, aves and mammals? Common to reptile, aves and mammals. Keratin found only in the body. Internal fertilization. Glands found in the skin. Presence of teeth. Aquatic habitat. Which of the following are common to reptiles, aves and mammals? Keratin found in the body. Keratin found in the body. Internal fertilization. Correct. Glands found in the skin. Aves no glands. Neither wrong. Presence of teeth. Aves no teeth. Wrong. Aquatic habitat. Aquatic habitat. Aquatic habitat. Reptiles. Aves and mammals. Following common. Reptiles, aves and mammals. Okay. A, B, E. Can go with A, B, E. A, B, E. Second one. Neither. Question number 11. Following characteristic features are found in animal class. Possess lungs for gaseous exchange. Ectothermic. Eggs without shell. Following characteristic features are found in animal classes. Possess lungs with gaseous exchange. Lungs with gaseous exchange. Ectothermic. Eggs without shell. Characteristic features are found in animal class. Lungs for gaseous exchange. In an animal class, ne? Enam first take a wrong. Ectothermic. No. Eggs without shell. Another characteristic. Okay. I am wrong. Let me check once again. Na? So this is all talking about one particular class. Ne? Possess lungs for gaseous exchange, ectothermic, eggs without shell. So this is amphibian. Ne? Another characteristic found in the above described animal class. Skin without scales. Fourth one. This is amphibian. Ne? Possess lungs with gaseous exchange, ectothermic. Because you know, ectothermic reptiles are ectothermic, amphibians are ectothermic, osteichthyes, chondrichthyes are ectothermic. And then egg without shell, you can't, then you can eliminate the reptiles. Lungs, then you can eliminate cartilaginous fish and the bony fish. So this is amphibian. This is amphibian. Few features of the life cycle of Pogonatum are given below. Out of the above characters that can be seen in plant life cycle of Nephrolepis are. Few features of the life cycle of Pogonatum are given below. Gametophyte is dioecious. Heteromorphic alternation of generation. Homosporous. Meiosis take place during spore formation. Sporophyte is fully dependent on the gametophyte. Out of the above characters that can be seen in plant life cycle, Nephrolepis. Gametophyte is dioecious. Monoecious, neither wrong. Again, monoecious means bisexual. Heteromorphic alternation of generation, yes, correct. Gametophyte visibly different, correct. Homosporous, correct. Meiosis take place during the spore formation. Meiosis take place during the spore formation. That is correct. Meiosis results spores, neither. Sporophyte is fully dependent on the gametophyte. Sporophyte is fully dependent on the gametophyte, no. So I would like to take B, C, D. B, C, D as the answer. 12th one, B, C, D. Third is the answer, ne? B, C, D. Question number 13. The life cycle of Neplephis differ from the life cycle of Pogonato. The life cycle of Neplephis differ from life cycle of Pogonato. In the presence of Antheridia, Neplephis differ from life cycle of Pogonato. In presence of Antheridia, in the presence of megasporangia, in the presence of microsporophyll, in the presence of sporophyte, in the presence of heart-shaped thallus, life cycle of Neplephis differ from Pogonato. Presence of Antheridia. No, both have Antheridia. Ne? Neplephis has Antheridia, Pogonato also have Antheridia. In presence of megasporangia, no, you don't see that in both. The presence of microsporophylls not present in both. Presence of sporophyte. No, sporophyte is present in both. In presence of heart-shaped thallus, that is correct. Fifth one is the answer. Heart-shaped thallus. Question number 14. Shown below is a life cycle of terrestrial plants. Life cycle of terrestrial plants. Which of the following responses indicate the presence of A, B, C and D in correct sequence? What is A? Gametophyte, female. Mitosis, neither. Mitosis. What is B? Zygote, sporophyte. Mitosis. C. Meiosis, ne? C. Meiosis. D. Spores, gametophyte, mitosis. If you take as events, neither. So let's take try to find the answer now. Meiotic gandavani, that's wrong. So if anything is present with meiotic, we'll reject it. Then mitotic division A, germination, zygote, no, neither. Zygote does not germinate. Ne? Mitotic division, growth and development. And mitotic division, germination, ekahari ne? Third looks okay. Germination, sporophyte, gametophyte, germination, no, wrong. Ne? Growth and development, germination, no, neither. No. So we have the answer, third one. We got the answer, third. Right, we'll start here, people. Structured essay. Right, structured essay part. I have given the marking scheme. Ne? Right, can you check the marking scheme, people? State what is phosphorylation. Shall I move to the marking scheme? Right. State what is phosphorylation. Production of ATP we call as the phosphorylation. Ne? Production of ATP we call as phosphorylation. V on the basis of the phosphorylation is divided as photophosphorylation, substrate phosphorylation and auxiliary phosphorylation. On the basis of the phosphorylation is divided as, okay. On what basis, Kilahanni? On what basis? According to the energy source. Following diagram shows mechanisms of enzyme action. Name and briefly describe it. So substrate, you go and bind. Active site is this shape. It's now modulating. Neither. So what is this actually showing, people? Induce fit. Induce fit. An enzyme is, now it briefly describe it. So here give a small description. name name induce fit. Then description, enzymes are not rigid structures. The interaction between the substrate and the active site may slightly change shape of the active site so that the substrate and the active sites become complementary to each other, right? Right, next one. State the difference between activator and enzyme cofactor. State the difference between activator and enzyme cofactor. Activators are regulatory molecules that bind to regulatory sites which stabilizes the shape. Now, activator and enzyme cofactor. Now, what do you know about cofactors, people? Non-proteinaceous components which are essential for catalytic activity of certain enzymes. Not all, certain. Activators are regulatory molecules that bind to regulatory sites which stabilizes the shape and the function of the active site. Next one, what happened to enzymes when the temperature is increased above the optimum? Hydrogen bonds, ionic bonds and other weak interactions are going to be disrupted. This result in change in the shape of the active site of the enzyme, which will alter the complementary nature of the active site of the enzyme. Therefore, complementary binding of the active sites and the substrate molecules will be prevented. Right, next one, following diagram shows two different types of inhibition. Now, here people, we can see the inhibitor come and bind with the active site and then prevent substrate binding. The other one, the inhibitor go and bind with somewhere else. So, one is competitive, this one is competitive, the other one is non-competitive. Hari neda. Ilangata. State how to identify two types of inhibition based on the substrate concentration. How to identify two types of inhibition based on the substrate concentration. Two types of inhibition based on substrate concentration. How to identify two types of inhibition. Now, what are those two types of inhibition? Competitive inhibition and non-competitive inhibition. Sorry, state how to identify two types of inhibition based on the substrate. Right, okay, what do you have to say that when the substrate concentration increases, if the inhibition reduces, it is a competitive inhibition. And in non-competitive, it does not respond to the substrate concentration. Right, part B. Following diagram shows some basic features of chordate. Identify them. A. Pharyngeal gill slits. B. Nerve cord. Neither. Nerve cord. Don't say spinal cord because the spinal cord is present in vertebrata. Right? But anyway, you can just say that people is a spinal cord. But anyway, the correct word is the nerve cord. Ilangata C is notochord. And D is muscular heart. E is ventral muscular, sorry, D is muscular tail. And then E is ventral muscular heart. Hurry. State 3 difference between chondrichthyes and osteichthyes. Skeleton is predominantly cartilage and bone. Heterocercal caudal fin, homocercal. Operculum present, operculum, sorry, no operculum, operculum present. Body is covered with placoid scales. Body covered with ctenoid or cycloid scales. Most show internal fertilization. Most show external fertilization. No swim bladder present. Right. State 3 structural similarities between about two groups of organisms. 3 structural similarities between osteichthyes and chondrichthyes. Aquatic, ectothermic, fins for locomotion, gills for respiration. Right, next one, following are features present in amphibia, reptilia. State whether they are present in amphibia, reptilia, or both. First animals to invade land. Amphibia only, neither. Reptiles didn't come for land first. Most terrestrial, some are aquatic and marine. Reptilia, why? Marine members are not present among amphibians. Thin body cover with scales, amphibia. Sorry, with no scales, amphibia. Internal fertilization, amphibia external, reptiles. Some limbless, both, neither. Both limbless members are there. Skeleton composed of bones, both. Right, moving to next question. Most of them have limbs for locomotion. State why amphibians and reptiles are said to be adapted for land life. Limbs available for both of them, possess lungs for gas exchange, their body cover with structures that can protect them from the environmental changes. Part C 1. What adaptation helps birds to fly? Light body, presence of wings, bones with air cavities, high metabolism, restriction in body size. State special features found, features only present in class aves. Special features only in aves. Front limb modified into wings with feathers. Having light and strong bony skeleton with air cavities. Beak without teeth, body covered with keratinized feathers. Legs have scales. Scales present on their legs. Right, state whether following statements are correct or wrong. Some mammals are adapted for flying. Correct, neither. Bats. Muscle diaphragm help for respiration of reptiles. Wrong, it's mammalian feature. Birds and mammals both have hairs for insulation. Wrong, birds have feathers for that. Complete circulation with four-chambered heart is present in both. Complete circulation with four-chambered heart is present in both. Makanda honey both mammals and correct or wrong. Complete circulation with four-chambered heart is present in both. State whether following statements are correct. Some mammals are adapted for flying. Both issue complete circulation with four-chambered heart is present in both. What is both? Birds and mammals. Put a doubt doubtfully. State whether following statements are correct or wrong regarding me. Nothing mentioned neither. And right, complete circulation with four-chambered heart present in both. Both I think it's referring to birds and mammals, but anyway, this question is wrong. I'll just leave that. I'll just leave it. Okay, question. Right, I'm moving to next question, people. State unique features of mammals. Unique features of mammals. Nourish young by producing milk with mammary glands. Having skeleton mainly composed of bones with cartilage articulating over surface. Body covered with hair for insulation. They have differentiated teeth. Muscular diaphragm. They have large brain compared to the other vertebrates. Have different methods of communication. Okay. Now we are moving, people, for second structured question. State C and E events. C and E. C and E. I think D. A. What is A, people? Sporophyte. Sporophyte. What is D? Gametophyte. B? Spore. No. B? What is B? Spore mother cell. What is B? Spore mother cell. What is C? C? Haploid spores. Sporophyte. C? Meiosis. Haploid spores. And D? Gametophyte. E? What is E? E. Gametophyte. Gametangia. Gametangia. I feel like that. What we'll see. Now, state C and E events. Okay, then I'm wrong. What is this? This has to be a dividing event. Then it is mitosis. Gametophyte mitosis. So, meiosis and mitosis. A sporophyte, B spore mother cell, D gametophyte, F zygote. What is heteromorphic alternation of generation? What is heteromorphic alternation of generation? Two multicellular body forms that alternate in life cycle of land plants are haploid gametophyte and diploid sporophyte, which are morphologically different. Therefore, called heteromorphic alternation of generation. What is the importance of carrying out internal fertilization for land plants? Prevent desiccation of gametes. What create diploid generation after fertilization? Delay of meiosis after the fertilization. So immediately after fertilization, meiosis does not begin. And then people marking scheme says fifth answer in seventh question. I'll come back for that. I didn't see this message. Sporophyte generation aqua adaptation need for successful colonization. What happened to gametophyte and sporophyte during evolution? Sporophyte become larger and the gametophyte become smaller. Sporophyte generation aqua adaptation need to successful colonization on the land and become dominant plant in the life cycle. Gametophyte generation has gradually reduced in size. Right. Part B. State three properties of Pogonatum gametophyte. Pogonatum gametophyte. Dominant plant. Large. Long-lived. Photosynthetic. Dioecious. Unisexual. State main parts of the sporophyte. Foot seta capsule. Pogonatum. Pogonatum. Then what is protonema? Green branch filament. State three difference between sporophyte and the gametophyte of Pogonatum. Sporophyte photosynthetic when young. Gametophyte photosynthetic. Diploid, haploid, short living, long living, reduced plant, dominant plant. Describe the following terms. Sorai, clusters of sporangia, fiddled young leaves roll lengthwise. Stolon, long underground branches arise from rhizome. Indusium, the structures which cover the sorai. State two difference between gametophyte of Pogonatum and that of the Nephrolepis. Gametophyte of Pogonatum and that of the Nephrolepis. Pogonatum gametophyte is stem, rhizoid and leaves available. Nephrolepis heart shape. Pogonatum gametophyte is dioecious. Nephrolepis monoecious. Pogonatum gametophyte Pogonatum gametophyte. We'll check this. Gametophyte is long living, dominant Pogonatum, not dominant. Similar to a plant and a thallus. Gametophyte of Pogonatum is dominant plant, but gametophyte of Nephrolepis is not dominant. Gametophyte of Pogonatum differentiated, not differentiated. Right. Next one, people. State how sporophyte of Pogonatum is different to sporophyte of Nephrolepis. Now, how the sporophytes are different. Sporophyte of Pogonatum not dominant, but dominant in Nephrolepis. Sporophyte of Nephrolepis is independent sporophyte. Pogonatum depends on the female gametophyte. Sporophyte of Pogonatum remain attached to the female gametophyte, but sporophyte of Nephrolepis is separate plant. Right. State 3 adaptations of sporophyte of Nephrolepis to land life. Nephrolepis sporophyte for land life. Cuticle is found in the aerial parts of the plant body. Right. Stomata are developed. 2 types of vascular tissue, xylem, phloem available. Vascular plants. Right. Now we come, people, for the essay part. First essay question, describe the Nephrolepis life cycle and state how it is more adapted than Pogonatum for land life. Describe Nephrolepis life cycle. So what are you going to see, people? Sporophyte is dominant, gametophyte is reduced and short-lived. Both sporophyte and the gametophyte are independent. Photosynthetic sporophyte have more complex structure. Plant body differentiated into root, stems and leaves. Cuticle found in the aerial parts. Stomata are developed on the aerial parts. 2 types of vascular tissues. They have fiddlehead young leaves. Stem is underground. Leaves are compoundly pinnate. Long underground branches. Stolons are there. Sporangia are developed as clusters called sori. And then sori are covered by indusium. When the sorus mature, indusium dry and exposing sporangia under dry environmental condition. Sporangium wall rupture and release spores. Spores are dispersed by wind. When the spores land on a favorable habitat, germinate. Gametophyte is small, heart-shaped. Rhizoid develop into ventral surface. Gametophytes are monoecious. Antheridium produces flagellated sperms, right? Gametophyte is monoecious. Antheridium and archegonia are developed on the ventral side. Antheridium produce sperms and archegonia produce egg cells. So this is then motile sperms swim into the with external water into archegonium. Sperm fuses with the egg after fertilization. Zygote develops into the embryo. All the development stages are nourished by the gametophyte. When young sporophyte develops, it is photosynthetic tissue become independent. Right, that's all. Now, life cycle details are simple. So, you learn the life cycle, you have to write those information. Now, people, the adaptations. What are adaptations? What is the question? Adaptations of Nephrolepis, then Pogonatum. Adaptations, then Pogonatum for land life. Right? Nephrolepis adaptations compared to Pogonatum. Sporophyte is dominant in Nephrolepis. Well differentiated plant body with stem, roots and leaves present in Nephrolepis. Pogonatum sporophyte does not have that. Presence of sporophyte attached to ground in Nephrolepis. Pogonatum, it's depending on the female gametophyte. Presence of adventitious root system. Pogonatum does not have root system. It has just a foot nedalamai. And process of xylem with tracheids to transport water. Process of phloem to transport food. Process of sclerenchyma and xylem. sclerenchyma and xylem to support the plant body. Presence of well-developed leaves and stomata. Presence of leaves with cuticle. Reduced desiccation. Right? Those features you can write. Those points you can write. Now we come to the second question. Some students have written only one question, the one essay. Now this is not a full paper, right? So the normal rules does not apply, right? So you write when two questions given, you answer both. Questions they can describe regulation of enzyme mechanism. Describe the enzyme regulation process, right? So what are you going to say, people? Now enzyme regulation happen on three ways, allosteric activators and inhibitors, cooperativity, and then feedback inhibition. You have to write that here. This is not regarding enzyme mechanism of enzyme action, right? So therefore, some students, when I check their answers in the class, right, they have written people regarding the enzyme mechanism, enzyme characters, so on like that. That means they have not read the question properly. So you have to read the question before you write, because the marks are allocated people for answering the question for not writing anything. Neither. So regulatory molecules bind to specific site, and we are non-covalent interactions and affect the shape, and it may result in either inhibition or stimulation. Most enzymes are regulated by allosteric regulation, each subunit composed of polypeptide chain, and then the entire complex oscillate between two different shapes, one catalytically active and the inactive. So once the regulatory molecule bind, then they bind with the allosteric site. So when activator bind, the stabilizes the shape of the active conformation, and when the inhibitor binds, stabilizes the inactive form. And subunits of allosteric enzymes arrange in such a way that the alternation of one affects for the rest of the ones, right? So when activator or inhibitor bind with the allosteric site, it not only affecting one, but it affects the all active sites. Right, and then ADP function as allosteric activator and ATP function as allosteric inhibitor. And right, cooperativity, second one, another allosteric activation, binding of one substrate molecule can stimulate binding or activity at the other sites. So there, the substrate binding to the active site, and that system is acting as a allosteric for the other enzymes, thereby increasing catalytic activity. Example, hemoglobin is made up of 4 subunits. The binding of molecule of oxygen to the first subunit increases the affinity of the remaining. Cooperativity work similarly in multi subunit enzymes too. Feedback inhibition is the last mechanism. Feedback inhibition. So in feedback inhibition, inhibitor bind to inhibitory binding of the end products to one of the enzyme involved in the process is going to reduce the reactivity. Feedback inhibition is an essential process for regulation. ATP exceed the demand of the body. ATP itself can act as an allosteric inhibitor, right? So those are the things that we have to discuss today, right? So thank you very much for answering all these 11 questions. I have to thank Neil, right? And then next paper is going to be 12th. Can you tell me the sections now for 12th one? So marking scheme says 5 as the answer is 7th question. I balum 7th එක 5 කියලා rate of the enzyme reaction මම මොකක් කියලාද ගත්තේ? මේකනේ වෙන්නේ. Seventh. Rate of enzyme reaction increase proportionately with pH and then become rate of enzyme reaction increases proportionally with pH and then become constant. Wait. Okay, most of the competitive which of the following statement incorrect regarding factors affecting rate of enzyme reaction? Most of the competitive inhibitors are irreversible inhibitors. Most of the competitive inhibitors are irreversible. Rate of enzyme reaction increase proportionately with pH. Metana make a wrong neither. Make a wrong lamai. Because this is for substrate neither. Rate of the enzyme reaction increases proportionately with the pH and then become constant. Him and neither. So I think I'm wrong here. But fourth one nega chutta balawala lamai. Fourth one. I think fourth one is also wrong. Ne? Fourth එක ළමයි බලන්න. What it says? Most of the competitive inhibitors are irreversible. Wrong name. Chuta balameka. Most of the competitive inhibitors are irreversible. Reversible. Allosteric activation. Oops. Temperature, pH, competitive. Most of these are reversible inhibitors. Most of the competitive inhibitors are irreversible. Reversible. Two wrong. Here, two wrong. Right. Lamai, seventh question ke hadaganna marking scheme maker. Answer is here 4 and 5 both. 4 and 5 both. Ne? Seventh question ke 4 and 5 both wrong. Ne? Okay. Right. Then that's all people. That's all for the day. Sections Q&A. What do you want me to right? What sections do you want me to revise and make fix questions? Now we have agreed to take plant life cycle. Ne? Plant life cycle, okay? Plant life cycle. I'll be taking people. I'll be taking I'll be taking Cetus, Selaginella, and Cetus. Tell me other sections. Chemical nature and function of main organic compounds. Chemical basis. Compounds. Domains. Domains can classification first part together. Can you suggest? Suggest, come on. I have to take plant life cycles anyway. Then diversity. Diversity. Codate we did discuss. Invertebrates we discussed, right? Fungi we didn't do. Fungi we didn't do, but we took some questions. And then people, how about evolution? Evolution evolution. It was a classification. Evolution. Evolution. Right? It's a plant life cycle, evolution. And then tell me from unit 2. Unit 2 again, cell cycle we discuss. Cell cycle we discuss. What else we took? Enzymes we discuss. How about photosynthesis? We did, the photosynthesis we did, but we didn't discuss it. How about cellular respiration? Isn't that good? Cellular respiration. Come on, tell me. Okay, Devmi, we took cell division. Karane api සෙල් විෂන් කරානේ ළමයි නැද්ද සෙල් විෂන් කරාද නැද්ද සෙල් ඩිවිෂන් we did නේ සෙල් සයිකල් කරේ නැද්ද did නේ අපි ගමු මොකද්ද දෙව්මි cellular respiration we'll rotate them we'll come back oh අපි rotate කරමු we'll come back for the same section some other time so we'll go on rotation now you know this is the 12th 13 and 14 එච්චරයි 15th paper onwards people we are taking animal form and function 2 එතකොට පොඩි combination එකක් තියෙයි නේද right okay then thank you very much for your participation හරි අපි එනං Saturday හම්බෙමු so until that නේද have some nice time right I'll see you all then later right okay then bye